Good day, this is the Professor, CBMD PhD, and today is Monday, August 22nd, 2022. It is 2:12 and 16 seconds, p.m. Eastern Standard Time, and today's research, sweeteners. So let's hop to it here. Non-nutritive sweeteners affect human microbiomes and can alter glycemic responses. Date August 19, 2022. Source: Cell Press. As in the late 1800s, non-nutritive sweeteners had promised to deliver all the sweetness of sugar with none of the calories, and they have long been believed to have no effect on the human body. But researchers challenge this notion by finding that these sugar substitutes are not inert, and in fact, some can alter human consumers' microbiomes in a way that can change their blood sugar levels. The researchers found that non-nutritive sweeteners affected the microbiomes of mice in ways that can impact their glycemic responses. The team was interested in whether these results would also be found in humans. And to address this important question, the research team carefully screened over 1,300 individuals for those who strictly avoid non-nutritive sweeteners in their day-to-day lives, and identified a cohort of 120 individuals. And these participants were broken into 6 groups, 2 controls, and 4 who ingested well below the FDA daily allowances of either aspartame, saccharin, stevia, or sucralose. In subjects consuming the non-nutritive sweeteners, we could identify very distinct changes in the composition and function of gut microbes and the molecules they secrete into peripheral blood. And this seemed to suggest that gut microbes in the human body are rather responsive to each of these sweeteners. When researchers looked at consumers of non-nutritive sweeteners as groups, they found that 2 of the non-nutritive sweeteners, saccharin and saccharose, significantly impacted glucose tolerance in healthy adults. But interestingly, changes in the microbes were highly correlated with the alterations noted in people's glycemic responses. And to establish causation, the researchers transferred microbial samples from the study, subjects to germ-free mice. The mice that have been raised in completely sterile conditions and have no microbiome of their own. The results were quite striking in all the non-nutritive sweetener groups, but in none of the controls. When researchers transferred into these sterile mice the microbiome of the top respondent individuals collected at the same point in which they were consuming the respective non-nutritive sweeteners, the recipient mice developed glycemic alterations that very significantly mirrored those of the donor individuals. In contrast, the bottom respondents' microbiomes were mostly unable to elicit such glycemic responses. And these results suggest that the microbiome changes in response to human consumption of non-nutritive sweetener may at times induce glycemic changes in consumers in a highly personalized manner. Researchers say they expect the effects of the sweeteners will vary person to person because of their incredibly unique composition of our microbiome. Researchers need to raise awareness of the fact that non-nutritive sweeteners are not inert to the human body as they originally believed. And with that said, the clinical health implications of the changes they may elicit in humans remain unknown and merit future long-term studies. In the meantime, we need to continue searching for solutions to our sweet tooth craving while avoiding sugar, which is clearly most harmful to our metabolic health. Drinking water seems to be the best solution. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.